Okay, cellular respiration part 2. We just finished glycolysis. That's the last thing I showed you. And I said that my video was long enough. And by the way, I said we're going on to part 3. This is obviously part 2. But anyway, we have to go through a step as large organisms to turn this 3-carbon pyruvic acid into something different. So what we're going to do is to come down into something called oxidative decarboxylation. Love this word because it makes me sound like I'm really smart because I know that word and I know I'm going to know what it means. For those of you that are seniors that had me, that was a bonus point on the test if you remember. I know you do. All right, anyway, what the deal is I need to turn this 3-carbon molecule into something that is literally going to be able to enter the Krebs cycle. Now, comment. We at this point are knocking on the door of the membrane of the mitochondria, but we're still in the cellular matrix, the cytoplasm. So what's going to happen is that our intermediate NAD is going to come in again, and it's going to grab some hydrogen. Now what happens when you grab a hydrogen, you grab a proton off of this, you're going to change the shape of it. You're going to change the conformation. When it did it this time though, before, up here, we never lost our 3 carbons. At this point, though, it knocks out a CO2. And we are left with a 2-carbon, what is called an acetyl group. All right? What the deal is, at this point, you've lost one of your carbons from your 3-carbon molecule. Of course, this same thing is happening to the other 3-carbon molecule. But anyway, so we've lost a CO2. So that right there, I'm going to take right here, and I'm going to circle our CO2. Our CO2 is gone, and I had this on black screen, so circling it with a marker didn't do much good. Our CO2 is gone, and so we're left with the 2-carbon here, and of course the other one that did the same thing. By the way, this is called oxidative decarboxylation, because if you remember correctly, this is a carboxyl group, and therefore we took it off. And oxidation reduction, do you remember what that means? Oxidation, and I'm not sure if we did this or not before I left. Oxidation, I always say Leo-Ger. If you lose an electron, the molecule is oxidized. And so we've lost electrons because we pulled them out. You gain electrons, then it's reduced. It's called, where you reduce the molecule, where you gain the electron. But on this one, we Leo'd it. In other words, we took out the electron, and when we did so, we took the carboxyl group off. So we decarboxyled it. That's what the word actually means. All right. At this point, we're going into what's called the Krebs cycle. Now, this is in larger organisms. Remember, our little yeast and our little bacteria don't need much. This is also known as the citric acid cycle because it's much better name for it since we're going to make citric acid. All right. This starts with, and at this point, we are in what is called the mitochondrial matrix. That's the fluid of the mitochondria. And I will show you a picture in the third part of this when we do the electron transport chain, what all that means. But anyway, at this point, we have our 2-carbon acetyl group. Now you'll notice I keep putting this in here because I'm going to have to do something to it. What the deal is that this acetyl group literally is going to attach with something called coenzyme A. Coenzymes are usually vitamins, so we're going to say vitamin A, but it's coenzyme A. And that coenzyme A is going to turn this into something called, it's a 2-carbon, acetyl-CoA. Do you have to remember all that? No, just the process. This is at this point ready to hook up with a 4-carbon molecule that is in the Krebs cycle, which is going to hook up, I'm going to put a plus sign there, which is going to give us our 6-carbon, and that is citric acid. That is one that I would suggest you kind of remember since this is the citric acid cycle. Now, all those other big names like the fumarate and the malate and all those other things, you don't really need to remember. At this point, and I'm sure that Sawyer or Samantha or Alex can tell you all this, anybody else that I had as a sophomore in honors biology, will tell you this is the green board. So what's going to happen is I'm going to get to a 5-carbon molecule. Now you'll notice we went from 6-carbon to 5-carbon. What must I have lost? Well, what you lost was CO2. All right, and so I'm going to circle this again. Out of our 3 carbons, we now have 2 CO2s that have come off. Okay, so we had that happen because, as before, NAD has swooped in, and I'm sorry if you can't see that, and has pulled out NADH. Now, some books you're going to see NADH2. In some books you will see NADH plus H. Doesn't really matter as long as you remember that this intermediary is coming in there and pulling off the electrons and the protons, and it makes our carbon leave. Guess what's going to happen next? We are going to come down to a 4-carbon. The 4-carbon at this point is going, it's because we've lost another CO2. So we've got our CO2. Let me circle it. We have now lost our original 3 carbons that we started everything with. We had the one way up here, the one right there, and the one right there. So all of our carbons are gone in the form of CO2. I'm sure you recognize that CO2 is what you are breathing out. So the food that you eat, you breathe out. But anyway, this also was done by NAD going in and then sending out NADH. Now, at this point, we're back to our 4-carbon that we started the cycle with, but it's not the same thing. So what we're going to do at this point is try and get this turned into that again. Okay? So what happens is this. We're still going to have our 4-carbon, but we're going to make some ATP. Now, I'm doing this very quickly because you've already gone over this, but ADP goes in and it grabs a phosphate group off of all this. It's still floating around in there, and we get ATP. There's more to it than that, not going there. So then at this point, what we're going to have is another intermediary that is called FAD. And it goes in and grabs a hydrogen and comes out as FADH2. Alright, so there's one. There's one. Oops, there's one. Alright, the last thing that's going to occur before I get back to my original 4-carbon is that NAD comes in one more time, grabs some hydrogen, and comes out as NADH. Once that occurs, this is that, and it's ready to pick up another acetyl group, and that's the Krebs cycle. Comment on several of these. Let me go back to the other picture, and let's say, for example, that you don't have glucose. Let's say you don't eat for a month. What's going to happen to your body? Well, your body is able to go to the liver, remember? We talked about that in cellular communication. That goes to the liver and will turn glycogen into glucose units. But let's say you use up all your glycogen because you're not eating. Eventually, your body will go to fat. But the problem is that fat doesn't necessarily turn into glucose. There are, evolutionarily speaking, we got to have some fail-safes. There are pathways down through here where different things can enter. For example, fats. For example, proteins. So there are fail-safes. So let's say that you have used up all your fat, you're skinny as a rail, then eventually your body will go to proteins, and that's when it starts feeding on your muscle and other tissues. That's when you've got real problems. All right? So there are fail-safes to get us to go on to this cycle. All right, on the very next thing that we're going to do, we are going to do the electron transport chain. For those of you, like I said, that had me as sophomores, you'll recognize the next one as the one that we had to draw. This one we did on the green board. The other one we did on the green board. The next one we're going to draw. That was part of your test. There's the end of that one. Part 3 coming up.